Hey everyone, in this video I'm going to talk about the changes to the number of chromatids throughout the course of meiosis. And keep in mind that the chromatid is the highly condensed coiled form of DNA, and a single chromatid would look like this. When it's joined at the centromere of another chromatid, they are referred to as sister chromatids. So let's see how the number of chromatids changes throughout meiosis. So during interphase, we should not see any chromatids at all. It should be in the loose chromatin form so that the DNA is accessible. After the S phase, the DNA is copied, and when those copied strands of DNA condense down into the chromatid form, which occurs during prophase 1, that is when you start to see these structures. So what we're going to say is that during prophase, we have double the number of chromatids you would have in a non-dividing cell. So throughout meiosis 1, the number of chromatids would stay double the amount until we get to cytokinesis. At the end of cytokinesis, the number of chromatids would be the normal amount. However, it would not be the full genetic complement. Here's why. So let's say we were taking a look at a cell, and the normal number of chromosomes in this cell are 2 homologous pairs, or 4 chromosomes. That would mean that the cell should have 1, 2, 3, 4, 5, 6, 7, 8 different chromatids after the end of S1, after the DNA has been copied. And these chromatids that you are seeing shaded in are the ones that absolutely must be present in order for the cell to function normally. So you could see that there's 1, 2, 3, 4 different shaded in chromatids that would be necessary for the cell. Well, at the end of cytokinesis 1, which is when we divide the cell like this, there are going to be 1, 2, 3, 4 chromatids present, but they're not the necessary chromatids needed for the cell to be functional. So yes, it has the normal number of chromatids, but the genetic information is not all present. So then during meiosis 1, the number of chromatids stays at normal amount until cytokinesis, which is when we're going to split this sister chromatid and this sister chromatid in half. So when these sister chromatids split in half, you get chromatids or cells with chromatids like this at the end of cytokinesis 2.